Good evening, everyone. Am I audible?
Yes, ma'am.
Yeah, thank you. Okay, let's start the session for today. Um, I'm Lavanya, a PMRF research scholar from IIT Madras. Today, we'll be having the week 7 live session for the course Tissue Engineering. So, the week 7 video contents are related to host integration and immune responses, key players in immune response, effect of biomedical implants, macrophages and its effect, and so on. Bioethics and tissue engineering. We would have seen about the ethical dilemmas and various concepts of bioethics. Finally, about skin tissue engineering. So let's move on to the first question. Dash arise from monocytes that have migrated out of the circulation and have proliferated and differentiated in tissues. Option A, macrophages. Option B, red blood corpuscles. Option C, osteoblasts. Option D, osteoclasts. So can anyone answer? What does arise from monocytes?
Is it macrophage?
Yes, yes. So the right answer is macrophage. So we would have studied about the hematopoietic stem cell, which will be present in the bone marrows. So hematopoietic stem cell will give rise to two types of precursor cells, that is myeloid precursor cell and lymphoid precursor cell in the bone marrow. That will then migrate to the blood. So here in the question, as we see, yeah, so macrophages will arise from monocytes. So these precursor cells will migrate to the blood and it will form monocytes, and this monocyte will move to the tissues and it will finally differentiate into macrophages. Question number 2. Dash is the process of particle ingestion. It is a cellular process of ingesting foreign particles and microorganisms. It involves three steps: recognition step, neutrophil attachment and engulfment, and third step, killing or degradation step. Identify the process. So the options are hematopoiesis, phagocytosis, apoptosis, and necrosis. So can anyone answer?
Is it phagocytosis?
Yes, it's phagocytosis. So phagocytosis, as we all would have known, it's a well-known process by now. Phagocytosis is a process of particle ingestion, basically. So the steps involved in phagocytosis are any phagocyte. First step is chemotaxis and adherence of microbe to the phagocyte. So a microbe or any antigen or any particle that is foreign to the cell is first phagocytosed. The pseudopods are formed, and this initiates the process of engulfment. Second step is ingestion of microbe by the phagocyte. So the engulfment process takes place. Third step is formation of phagosome. Phagosome or otherwise called phagocyte or phagocytic vesicle is formed around the microbe. Fourth step, phagolysosome formation. So lysosome, as we all know, is a cellular organelle which contains digestive enzymes called lysozymes. The phagosome and the lysosome fuses together to form a phagolysosome. Fifth step is the digestion of ingested microbe by enzymes. So the microbe will be partially digested, and the indigestible material will also be present and will be eliminated from the cell in residual bodies. So formation of residual body containing the indigestible material will be the sixth step, and finally discharge of the waste materials from the phagocyte. So these are the overview of the steps that will take place in a phagocyte. Moving on to the third question. X are endogenous equivalent of pathogen associated molecular patterns, PAMPs. These are released by necrotic cells to signal tissue damage and are recognized by macrophages and dendritic cells via pattern recognition receptors, that is PRRs. So examples of X include uric acid, ATP and heat shock protein. So what would the X be? Any answers? Okay, so the X are alarmins. So alarmins are endogenous equivalents of PAMs. So whenever a cell, a necrotic cell or an activated immune cell, whenever it is under the influence of any stress, damage, injury or even due to age, it will release signals called alarmins. So examples of alarmins are uric acid, ATP and heat shock proteins. These alarmins will then be recognized as antigens by the dendritic cells and other cells such as macrophages, that is mentioned in the question, macrophages, endothelial cells or leukocytes. So these alarmins will be recognized as antigens by these types of cells, and when it is recognized by a dendritic cell along with the MHC complex, major histocompatibility complexes, it will be processed and presented to the T cells, and the T cell receptor, once the antigen is presented, the T cell receptor will undergo, T cells will undergo polarization. So has a if any T cell undergoes polarization, it will be just a second. Yeah, sorry. So T cells, when it undergoes polarization, it will be differentiated into effector cells, cytotoxic T cells, helper T cells and regulatory cells. So as we can see here, T cells and Treg cells are formed. T cells are for amplification of the inflammatory process, and Treg cells are for tissue repair and protective effects. So when any cell is in the necrotic stage, these processes takes place. Moving on to the next question. MMP9 is a key enzyme in the degradation of collagen implants with a combined action of gelatinases and collagenases. Increased levels of MMP9 are indicative of inflammation and are associated with poor wound healing. Are these statements correct? So are increased levels of MMP9 responsible for poor wound healing process? Can anyone answer?
Yes.
Yeah, correct. So MMP9 is an example for collagenase enzyme, and so whenever a wound healing process will occur, one of the main step of wound healing is the collagen deposition in the wounded site. So MMP is since it's an example for collagenase, it will inhibit the formation of collagen. So as an example, I have given here in the figure So during a myocardial infarction is a disease that takes place in the heart. So during any myocardial infarction, there will be release of DAMPs, that is damage associated molecules, molecular patterns. So DAMPs will be released. So examples of DAMPs are box1, heat shock proteins, fibrinogen, fibronectin, ATPs, HAs, and other complements. So these released signals will recruit cells like macrophages and neutrophils, which will release pro-MMP9. So these pro-MMP9s will get activated to form MMPs, and MMPs will then lead to the will then activate CDs. So CDs are a cluster of differentiation, which will be ligands present on the cell surface. So these MMPs will activate CDs, and other genes such as CXCL6, and it will also induce the formation of IL-1 beta, IL-8 platelet, and glycoprotein 4 formation, which will then finally be responsible for the inflammation process. So MMP, yes, it is a key enzyme in degradation of the collagen, and it is also an indicator for inflammation. So TIMP, which is a tissue inhibitor of matrix metalloproteinases enzyme, when these are the enzymes that inhibit the MMPs, and all these enzymes, so we need a balance between the MMP and the TIMP for tissue repair and rebornylation processes to take place. Moving on to question number 5. Comfeel is a dash dressing that promotes debridement and angiogenesis. It does not adhere to the wound bed and is useful for low to moderate exuding wounds. So option A, alginate dressing; option B, hydrocolloid dressing; option C, foam dressing; option D, semi-permeable film.
Hydrocolloid dressing.
Yes, so comfy feel is an example for hydrocolloid dressing. Hydrocolloid dressing is nothing but the it is a formation of a gel. So polymers will be used mostly, carboxymethyl cellulose and gelatin. These polymers will be frequently used to form a gel, which is nothing but addressed as a hydrocolloid dressing. This Yeah, this Comfeel is a patented product, which is an example of hydrocolloid dressing, and it is used for low to moderate exuding wounds. So, since exuding wounds means in a wound site, there will be a lot of wound exudates. That is the pus and the fluids that is being released from the wound site. So this will be able to absorb all those wound exudates. Moving on to question number 6. Which one of the following belongs to the class of third line of defense and a specific adaptive immunity? So option A, mucosal membrane; option B, antimicrobial substances; option C, antibodies; option D, microbiota. So which one of these the following options belong to the third line of defense?
Antibodies.
Yeah, correct. Antibodies. Yes. So option C, antibodies. So sorry. So the first line and second line of defense come under non-specific defenses, and third line of defense come under specific defenses. So antibodies will only be produced from the B cells towards specific antigens. So the first line of defense includes skin, mucous membrane, and secretions from the skin and mucous membranes. Second line of defense include phagocytic leukocytes, antimicrobial proteins, inflammatory responses, and fever. So the third line of defense include lymphocytes, antibodies, and memory cells. So question number 7. A scaffold is fabricated by a team of researchers. In vivo testing has been performed in animal models. Within a few minutes of implantation, blood proteins can adsorb to the biomaterial surface that may lead to host immune reactions. Which of the following mechanisms can activate this immune response? So option A, complement system activation. Option B, coagulation cascade reaction. Option C, YAPTAZ. Option D, erythropoiesis.
Option A and B.
Yes, that's right. So option A and B are the processes that can activate the immune response, initial immune response to any biomaterial surface that is implanted, and that is blood contacting. So these are the two options. Question number 8. Which of the following are example of alarmins? Yeah, so as we saw in the previous question, uh, select one. So since one option is the correct, yeah,
Uric acid.
yes, correct. So uric acid is one of the example for alarmin. Question number 9. What are the 3 R's in animal research? Select one option. So option A, reuse, refine and replace. Option B, replacement, reduction and refinement. Option C, repair, regenerate and reduce. Option D, regeneration, reduction and redefinement. So what is the 3R principle that is used in animal research?
Option C.
No, the answer is option B. So it's replacement, reduction and refinement. So what basically these three terms mean is that replacement. So an animal experiment. So when we are using animal experiments, there might be other alternative methods also by which we will be able to test our material. For example, when we are preparing a cosmetic that will be used for our human purposes, uh, it is not it is not very necessary to use an animal to test those unless and until it is toxic for the skin. So or if we are using any material or any hydrogel or any solution that is life saving will be given first preference for using animals and in vivo testing. So an animal experiment is only approved if no other suitable alternative method exists, such as computer simulation, cell culture experiments and so on. So if you just want to see if the material is toxic or not, then maybe you can just see it in cell culture experiments. Or if you are able to screen and if you are able to get the results using computational methods, then only that method will be preferred first. So if none of these methods will provide an answer, then we will go for animal experimentation. And reduce is for using small number of animals in our experiment group. So the number of animals that we are using per experimental group will be determined by a process called randomization. Randomization also won't be done by us. We won't fix the number of animals by our choice. There are specific softwares to randomize how many animals are required to be in a group and how many groups of animals are required to carry forward our particular research. So we have to choose the smallest number of animals necessary for our experiment to and the smallest number of animals that will produce significant statistically significant results. And finally, refine. So housing and experimental conditions are will be constantly maintained and optimized for all the laboratory animals in order so that it will experience only little amount of stress. So since we are using animals for our study, we should not put them in a very stressful environment. So always we have to keep checking the animal house conditions to maintain little stress environment. So this will include animal friendly experiment methods, promoting non-invasive procedures, and treating any pain the animal may be in. So animal friendly experimental methods means, for example, if it is a, so like I said, if it is just a cosmetic material, yeah, we shouldn't what to say, promote the formation of blisters or anything on the animal skin so that the to test the cosmetic material. We can just see if the cosmetic material by itself is toxic to the skin of the animal. And promoting non-invasive procedures. So for wound healing applications, we will just prepare an excision wound model that is superficial for the animal, and it will not induce the death of the animal, and it won't be very invasive also, it will be minimally invasive. And so we would have heard about injectable materials. So injectable materials also very minimally invasive procedures, I mean biomaterials, hydrogels that can be tested for their application, and treating any pain the animal may be in. So if the animal is in a lot of stress, then we will be forced to euthanize the animal. Or if the stress is manageable, if we can provide any treatment for that pain, like providing antibiotics, timely provision of antibiotics, then we have to follow that also for the animals. 10th question, which of the following is synthesized by fibroblasts during the process of wound healing? So what is the one particular component that will be synthesized inside the fibroblast cells during wound healing? Procollagen, hemoglobin, thrombin, myosin.
Thrombin?
No. The So the molecule that is synthesized is procollagen. So whenever a fibroblast, so during wound healing, there will be the fibroblast cells will be activated first, and it will be activated to synthesize procollagen chains. So in the nucleus, the collagen mRNA will be transcripted, and it will be translated in the rough endoplasmic reticulum, where in the rough endoplasmic reticulum the pro alpha chain backbone of the procollagen that will be formed first, followed by hydroxylation of that chain. So hydroxylation will particularly occur in proline and lysine residues, and then glycosylation of the chain will occur. So this is the procollagen that is formed. It is a triple helix structure, and this procollagen will be exocytosized by the Golgi, and then in the extracellular space, the procollagen will be cleaved to form a tropocollagen, and finally the formation of collagen fibers. So the tropocollagen that is formed will be cross-linked by lysyl oxidases to form stable collagen fibers. So all these till the step 4, it will take place inside the fibroblast cell, and after which it will take place in the extracellular space. So some questions from the previous assignment, the 6th week assignment questions. Which of the following extracellular matrix protein has RGD domain? Option A collagen, option B thrombospondin, option C entactin, option D all of the above. So which of the following will have RGD domains? Can anyone try answering?
D.
Yeah, it's correct. So RGD domains will always be present on proteins. So here collagen, thrombospondin and N-tactin will are all examples of protein polymers. So it will have RGD domains in them. Selectants are Cl minus. So sorry, that's not Cl hyphen. It's Cl minus dependent binding proteins expressed during repair phase and involve in inhibition of neutrophil migration. Is it true or false?
A.
No, selectants are not Cl dependent. So in the last class, I would have in the last class I would have explained about adhesion proteins. So selectants are basically Ca dependent binding proteins. Consider question number 3. Consider the migrating cell that is shown in pink color and brown color in substratum. Here, which is the leading edge, A or B, and is it called what it is called? So there are only two options. Which is the leading edge, A or B, and the name of the leading edge, Europodium or Lamellopodium.
Is it option C?
Yes, so A is the leading edge as we are able to see in the figure, and it is always called as Lamellopodium. Which of the following signaling molecule that control the generation of tissue formation, mitogen or morphogen?
Option A.
Uh, option A. So the answer is option B.
Yes.
So morphogens control generation of tissue formation. Mitogens will control cell division. So the option is morphogen. What conditioning types are applicable for cartilage cell culture in a bioreactor? So what are the conditions that should be maintained for if you want to culture a cartilage cell or
Option
tissue
C.
in a bioreactor? Sorry, option?
C.
Yeah, option C is sorry. Option A and B is correct. I'm sorry. The options here, sorry, the answer mentioned is wrong. The correct answer is option A and C, compression and shear. So since it is a cartilage cell, cartilage or a bone cell, whatever we culture, the main conditioning types that is required is compression and shear. So since these two types, cell types have the highest mechanical strength, these are two conditioning types that are required. Electrical will be used for muscle tissue, heart tissue, and so on. Question number 6. Match the following. So we have to match the type of migrating cell to the role of the migrating cell. So options are option types of migrating cells are neutrophils, endothelial cells, tumor cells, and fibroblast cells. Role of migrating cells are metastasis, wound healing, angiogenesis, phagocytosis of bacteria.
Ma'am, is it last option?
Oh, yeah, last option.
Yeah, that's correct. So option D is the right answer. So first, neutrophils are, the main role of neutrophils is phagocytosis of bacteria. B, endothelial cell. So the main role of endothelial cells is angiogenesis. So whenever there is a formation of new blood vessel, the major cell type that is present will be endothelial cells. Tumor cells will, the role of tumor cells is metastasizing. And finally, fibroblast cells, which will be involved in wound healing. Yeah, so these are the questions for today's session. If anyone are having any doubts, you can ask.
Hello, ma'am.
Yeah. Yeah.
Ma'am,
Hello.
like, yes.
Yes.
For the last assignment, there
Yeah.
are questions like puzzle solving and crossword and all.
Okay, this is for the week 6. Is it for week
Yes.
6?
No, it is for week 7.
Okay, this week's, okay, fine, yeah.
Oh, no, week 6 only. Sorry.
Okay, yeah, yes.
Will such questions also come for the exam, main exam?
Yes, ma'am, it will come.
Okay.
So the main exam, I think the exam is on September 24, if I'm not
Yes.
wrong.
Yes.
Yeah,
Okay.
so for the main exam, so this, since this is an online exam, there will be questions like this, and there won't be any big questions. So just 1 mark or 2 mark questions only, mostly.
Okay. Thank you.
And in the previous session, someone asked if there will be any essay type questions.
Yes,
So no,
ma'am.
here no essay type questions will be there. Only
Okay.
only the questions that you are um, facing in the assignments, every week assignment, only questions, those type of questions will be there.
Okay, ma'am. Thank you, ma'am.
Okay, yeah. So if there are no doubts, then we can end the meeting here. Thank you all.